This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 295, recorded on July 25th, 2014. Hello, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today right here in the TWiV studio is Dickson Despommier.
Hello, Vincent.
How are you today, Dickson?
I feel great.
Do you?
Yep.
There's nothing at all wrong with you?
I just got my first set of glasses yesterday.
Where'd
I
you
went
get
to the
them?
eye doctors because I was having vision problems in my right eye.
Mm-hmm.
And lo and behold, I found out that through an old student who's now practicing ophthalmology in our eye institute, he told me, you know what? You've got a stigmatism in your right eye, a big one, and in your left eye, you've got a small one.
Did they take them out?
No, they don't do that.
You said
This
you-
is an irregularity in your cornea.
And what did he do to fix it?
They give you glasses, just like you. You have glasses.
Did you get them yet?
I have the prescription filled. They won't be ready until this coming Friday. But the moment he put in the correct diopters and said, "Can you read that?" I mean, it was like a door opened, and I could see again. You know, I felt like I was going blind or something, and of course I wasn't, but I felt as though I was.
All right, well, let's keep that door open, Dickson.
You betcha.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
Hi, Rich.
Hello, Rich.
How are you today?
Dandy.
It's Friday.
Real good. Yeah, Friday's good.
right.
I'm headed for Amelia Island this weekend to mooch off my brother.
Nice.
Amelia Island, is that
Yeah.
in North Carolina?
No, that's the northernmost barrier island in Florida. It's just 2 hours away.
Oh, wow.
That's nice.
Hang out on the beach. You
Do
know?
any fishing?
No,
You're going to sail?
no.
Okay.
Gonna veg.
You got a drink?
No. None of the above.
Well, he's got to
It's a-
stay hydrated,
That's
Dickson.
true.
veg.
No,
Yeah, I'll do
Needs
that.
like water.
I'll
and
drink
stuff
some
like that.
water.
Oh, good. Okay.
You don't want to be dehydrated.
And you need to know that it is 88 degrees Fahrenheit here, 31 Celsius. Humidity, for those who are interested, is 65%, and it's partly cloudy. It's a beautiful summer day in Florida. It would kill most people from the north, but it feels fine to me.
Right.
Also joining us today from the north, up here, Western Massachusetts, Alan Dove.
Good to be here.
Would you
Alan.
die if you went to Florida?
No, we had weather like that just a couple of days ago.
Yeah, we
here.
did.
Yeah.
We did.
It's
Yeah,
hot.
and now it's currently 81. Absolutely gorgeous, clear skies. It's supposed to be nice tomorrow, and then thunderstorms on Sunday.
And the
Here.
humidity?
Humidity is 42%.
Thank you.
We are 27 Celsius here in New York City, 41% humidity,
Right.
and it is sunny. There are no clouds.
With a slight
Yeah, a
breeze
few
out of
clouds.
the southwest.
We are privileged, Dickson and I, to have a view of the Hudson River.
We are.
Well, we have to stand up because they didn't.
Yeah.
We don't have ceiling to floor windows, unfortunately. But
That's true. But we
it's out there, right?
It is out there.
And we can see New Jersey, the George Washington Bridge.
Yes.
We
indeed.
can look south.
We can, and we can see the weather patterns coming towards us.
When it storms, it's quite spectacular, isn't it, Dickson?
And we have some amazing sunsets here, too.
We do. And that's it for today.
Yep.
But
See
anyway,
ya.
it's a beautiful day.
Take it easy, guys.
All right. We have
Hey, wait.
2 follow-ups, or is it follows up? Alan?
Follows up.
Follows up.
It's like brothers-in-law.
Whoppers Jr. Yeah.
Okay. I pulled a comment off of Twitter from Stephen Goldstein, who said, "Thank you for your latest defense of scientific freedom on the most recent TWiV. We are here to defend your freedom to do science in a responsible and safe way, right?
Right.
Exactly.
We do want a reasonable discourse, and we do think that experiments are important to do. I was thinking this morning, there are some experiments I wouldn't want done.
I was
Sure.
thinking about the same kind of thing.
If you wanted to make HIV airborne, transmitted, I would say let's not do that.
Yeah.
Because it's out of the realm of the biology of that virus, and you're not going to learn very much, probably. Although, again, who knows? You could learn fantastic things. But on the other hand, aerosol transmission of influenza is part of the biology of that virus. So if you have a strain that doesn't transmit and you want to convert it, that makes sense to me.
Yeah.
What about you, Rich? Is there any experiment you would not want to do?
The first sorts of things that come to mind are, there was a series of experiments done a while ago with sticking cytokines into various pox viruses. This was one of the first things that came up on the radar as dual use of concern type experiments.
Yeah, yes.
This particular virus was able to successfully infect animals that were vaccinated against the same agent.
That's right.
So you're talking about a pox virus that can break through vaccination.
Yeah.
So I wouldn't want to, say, engineer smallpox to express interleukins.
Yeah, yeah.
Right.
I just think that might be stupid. That's kind of an extreme example, but
Sure.
I'd have to think to come up with some others.
Yeah, we talked about that a long time ago. That was done in Australia, that experiment. Yeah.
But we do engineer viruses to carry things for us into certain cell types like cancer cells.
We do.
And that's a gain of function study of sorts, and it's directed towards the cancer cells. But what if, by some quirky chance, the virus instead invaded all the cells?
This is why experiments like this have to take appropriate safety precautions. You have to figure out,
of course,
kind of what the possibilities are, and take appropriate safety precautions. And in citing that experiment that I did, I'm not arguing that the initial experiment shouldn't have been done. There was actually, although it was debated, there's actually a logic to doing that experiment, because you want to try and use these things as vaccine vectors, and by putting cytokines into them, you might alter the immune response in a way that makes the vector better. And in fact, this kind of thing is being done with oncolytic vectors. Yeah, sticking in cytokines to tweak the immune response to the vector and enhance the immunity to the tumor. Sure. So in general, the experiments are reasonable to do, but there are limits to how you might want to explore it.
Yeah.
Well, you can imagine scenarios for many experiments, right, Dickson? But you cannot. I mean, you were talking this morning about how things could happen, but it could happen in all walks of life. We sent people to the moon, and that was dangerous. They could have all been killed. But you cannot let fear paralyze you. You have to sometimes do scary experiments. And I think if you have the right containment, then you're doing your best. And actually, our next follow-up.
So if I could just interrupt
for
one moment and say that we made a conscious effort to ban biological warfare. And part of that is piggybacking viral genomes on top of each other, so that you've delivered 5 or 6 different pathogenic viruses all at once, or you've made it more pathological for people than it used to be. All of those for the purpose of infecting people. Those kinds of things are horrible. And I would certainly say we shouldn't be doing those things. Although
warfare for killing people, we don't condone that, because we're not learning anything.
Yeah, but in fact, I think the only category of experiments that I would outright say, that should be banned, would be ones that are deliberately aimed at making pathogens more pathogenic in people.
Yes.
If you want to find new ways to grind up anthrax, weaponize anthrax and distribute it, well, no, we shouldn't be doing that work.
Right.
But in terms of what Rich cited, the cytokine experiments, that's edgy, and that's something that I would hope people would think about very carefully before doing it.
Sure.
Although those experiments did have a goal,
and they had a goal, and you could also look at, well, are there other systems you could do this in to kind of sneak up on the answer? Could you do this in monkeypox? Could you do this in something else that's not variola? There are ways of approaching questions that may not be quite as dangerous if it really freaks you out. But I'm not sure that even that is something that I would say outright shouldn't be done.
There are many ways that you could modify a virus so that it would even be more powerful at evading immune responses, because we know a lot of modifiers from different viruses. We could combine many of them into one virus, for example. Nobody proposes these because you're not going to learn anything from them. The point of those is to make a killer virus. And so that's what always has been my philosophy, is that a good, inquisitive, trained scientist is going to design experiments to learn, not to injure. And if there is a possibility of injuring, I think we just need to do the proper containment. But the experiments are worth doing. We should do them.
Right. And so I've actually asked someone who does run a high security facility close by where we are just recently whether or not he could think of any viruses or bacteria, for that matter, that got loose from a laboratory that caused a pandemic, and he couldn't think of any. Of course, there haven't been any. So,
which is not to say that it can't happen. But
that's right. The fear of it happening is one thing, but actually saying we're going to prevent this from happening because we're afraid it might happen, that's yet something else. And so what he was expressing was a mistrust of the security systems that have been created, no matter how secure you say they are. And that's their premise. I mean, the premise of the fearmongers, I call them fearmongers because I'm allowed to say that, are that we're careless people. And look, here's some proof of that. Lots of stuff that we found in refrigerators and freezers and stuff, but none of that ever resulted in an outbreak of anything. The outbreaks occur from nature,
or at least not an outbreak that went anywhere.
Exactly. Even when germ warfare was being used,
yes,
it did not result in pandemics.
Right.
So,
how did we get on this? Oh,
no.
Containment.
Containment and carelessness in the laboratory. There's no question.
So
but
this is not just continuing last week's discussion. This is going to lead into a cool paper.
Yeah, we had a follow up from last week's discussion from Ryan, Ryan was or still is a postdoc with Ben Tenuver, who is at Mount Sinai here in New York, and who we've had on the show. He writes, Vincent and the TWiVome, fantastic discussion last week, TWiV 294, on biosafety, security, gain of function work, etc. It appears that this debate isn't going away anytime soon. I thought I would share our strategy of molecular biocontainment, where target sites for microRNAs expressed in humans but not experimental models are inserted into influenza. And he published this in 2013, Nature Biotechnology. This system would then prevent spread from the laboratory into the human population. While not suitable for all situations, this strategy could potentially offer a solution to some of the concerns being raised. I would love to hear the TWiV team's thoughts.
That's interesting.
Best regards, Ryan. Yeah, so he had actually mentioned this to me a few times before, and for various reasons, I suppose, we never got to it, but now is actually a perfect time. This is a paper in Nature Biotechnology. It's called MicroRNA-Based Strategy to Mitigate the Risk of Gain-of-Function Influenza Studies. And Ryan is the first author, and it includes members of the Tenuver lab, members of the Garcia Sastre lab at Mount Sinai, and also members of Daniel Perez's lab at the University of Maryland. And so let's discuss this. This is really interesting, and whether this might help mitigate some of the concerns. And as everyone knows, this is sort of catalyzed by the avian H5N1 influenza virus experiments, which we discussed a lot on TWiV, where we know that these don't transmit well, if at all, among humans when they infect us. So a laboratory asked, what does it take to transmit between ferrets, another mammal? And they selected for strains that could transmit among ferrets. And that started this whole brouhaha about dual-use research of concern, gain of function, and whether these strains would infect people if they were released and so forth. And we're still debating this to this day. So their approach is, can we build something in to these viruses, H5N1 or whatever virus you want to study in an animal, that would prevent it, if it got out, from replicating in people. And microRNAs are perfect for this. To review a little bit about microRNAs, these are small RNAs produced in cells. They have regulatory roles. So they are derived from longer precursor transcripts. They are processed to 21 nucleotide long microRNAs. And then they hybridize to a target sequence in an mRNA, for example, and lead to its degradation or inhibition of translation. So the idea would be to put a microRNA, say, into H5N1.
Target site.
Target site, so that say the microRNA that would bind to that target site would not be present in ferrets or mice, but would be present in people.
Humans, that's right.
So it would prevent its replication. Okay? So they searched for microRNAs that fit this criteria. In other words, they're abundant in human cells, and they looked in a human lung cell line, but absent in ferret cells and in dog cells. So
both ferrets and dogs being related, at least somewhere on the phylogenetic trees. They're both carnivores.
Yeah,
right. They're both in the order Carnivora.
And
I think one of the reasons they needed to do dog cells is because they need a cell culture host to grow these things, and they use canine kidney cells.
Right. The canine MDCKs are commonly used to grow influenza virus strains. So they started by simply isolating small RNAs from these human lung cells, from MDCK cells, from primary ferret lung. They clone them, they deep sequence them, and then they simply ask, can we see some by sequence and by abundance that are in ferrets and dog cells, that are not in ferret and dog cells, but are in human cells? And they could easily find 3, I think, initially. They're miR138, miR193B, and 192. They then did northern blot analysis to verify this restricted expression, and that narrowed it down to 138 and 192. And then in the end, they looked at PCR by PCR at these and found that 192, 1 microRNA, is found in human cells. It's produced in human cells, but not in ferret cells or in mice. Now they started looking in mouse cells as well. So miR192. So far we have an observational study where we've identified that ferrets and mice do not produce this miR192, but human cells do.
Yeah, you want it in mice so that you can use the mice as in a way, a surrogate in vivo model for humans, ultimately.
Right. Yeah, you can use ferrets, you can use mice. People use both.
But the ferrets don't have it, right? That's one of the points. Ferrets don't have you need an animal model that expresses the microRNA, so you can do the final experiment to show that the virus is not going to replicate in that animal.
Right.
You need a non-permissive, a host that should be non-permissive.
Right.
So then they introduced the target for miR192 into the HA RNA segment of influenza virus. They do a little engineering so that they minimally affect this segment, and they produce a virus which now has the target for miR192 in the HA gene. And this replicates, apparently, as far as titers go, pretty well in MDCK cells. So MDCKs do not express this 192. The virus replicates quite well with or without the 192 target inserted. And if you now express the microRNA in MDCK cells, you can do that artificially. The HA levels are down. And then if you put this virus into A549 cells, human cells, which produce 192, the HA levels are reduced and the virus titer goes down. So with normally the titer would be between 10 to the 6th and 10 to the 7th PFU per cell. And if you put them into these, if you put them into 192 producing cells, the titer is reduced to 10 to the 5 PFU per cell.
Yeah, in order to find the human cell stuff, I don't know why they did this. You got to go to the supplementary figure.
Yeah, the one they show is the MDCK
Right, and the supplementary figure shows they don't have as many points on the A549s, and it's not quite as dramatic a difference. It's only about a log, but it's still a difference.
Yeah, this is another reason why I despise supplementary figures, because, who's to decide what you need when you're looking at the paper, and going back and forth I find is annoying. So they call this blunting replication. That's an interesting term.
I kind of liked that term, actually. It's
it works.
The first
it's
time
fine.
I've seen it works, yeah.
But it's not zero. So in the presence of the microRNA, you don't completely block replication.
Right.
And that's interesting. So they actually pursue that a little, and they ask, is that because you're getting mutation of the target site? And apparently it's not, right? So this is, even with the microRNA present, you can still get some replication, but it's much less. And then you'll see that makes a difference. So if you take this and put it in mice, so now you infect mice intranasally, and the mice are making 192, and you put wild-type virus or the 192 containing virus into mice. Wild-type virus, in terms of weight, the mice lose weight very quickly. Within 7 days, they lose 75% of their weight. And this is a typical assay for influenza virus pathogenesis. But the 192 target containing viruses do not cause any weight loss, even when you put 10 times more virus into the mice. So it's interesting that and this if you look at the viral titer in mice, it's reduced when you have the miR192 target site. It's not abolished completely, sort of similar to what you see in cell culture, but it's enough to prevent pathogenesis.
Yeah, and the survival curve is pretty dramatic too.
Yeah, so that all the mice survive.
All the mice with the control virus are dead in a week, and the ones with the 192 virus are doing fine.
All of the blots and stuff that they show gene expression from the virus also indicate that the replication is knocked back very considerably. In fact, of all of this, the wimpiest data are the growth curves. Okay, and they are in fact multi-step growth curves, which has a tendency to exaggerate some of these effects. I'd wondered what single-step growth curves would actually look like, but it doesn't matter to me. It's the mouse pathogenesis. First of all, they're showing the principle works, and secondly, the pathogenesis in mice experiments are dramatic, and it in fact emphasizes that if you screw up the virus even just a little bit, you can have profound effects on the pathogenesis. That's an important principle in general that comes into play in all of this gain of function stuff.
Yeah.
So then the last experiment is, they say basically these look nice, but putting in the microRNA could screw things up, even though in a growth curve it looks fine and so forth. So they ask, do they transmit in ferrets? So ferrets are not making miR192. So you're basically looking at whether the sequence that you've added, the target, is affecting transmission or not. So they do transmission studies with a seasonal H3N2 strain, which is known to transmit among ferrets, and they put the miR192 target site into the HA of that virus. And they basically show that this is transmitted as you would expect. There's no difference with and without the miR192. So apparently that is not affecting any aspect of the biology. So that's good, because then you could make the argument that if you wanted to do transmission studies in ferrets, you should insert this miR192 sequence into the virus.
Yeah.
Now, of course, you can never argue out of the fact that it might be doing something, right?
Right.
But I think for at least initial studies, it wouldn't hurt to have this present. And what do you guys think? Is this something that people should put into their viruses when they're doing gain of function?
Well, the first thing I think is that just, regardless of its utility, the whole set of experiments is cool.
It's very cool.
Yeah.
Absolutely.
That you can dream up this idea about how to use microRNAs to manipulate this, and have it play out in the experiments is very cool. Yeah, I think that this is a perfectly reasonable way to put a level of biocontainment on viruses to do the experiments. I kind of like it.
Yeah, I think it would be especially useful if you were doing, sort of a first round experiment. Gee, what mutations would increase transmissibility, for example? And you're passaging the thing through ferrets or whatever other a host that you're working with, probably ferrets in the case of flu. You don't know what you're going to get out of that. So you could put something like this in just as an added safety precaution, see what you get, what mutations improve transmissibility, and then go kind of more surgically and insert just those mutations, maybe in the absence of this 192, and see if those are indeed causing the phenotype. But that way, more of your work would be done with this potentially safer virus.
Yeah, it's kind of like sticking the H5 hemagglutinin in a safe background, okay, and doing experiments like Kawaoka did to start with. There's a bunch of different ways to do the preliminary experiments in a safer environment, and then as proof of principle, and then do a higher containment stuff to verify your results.
Yeah, especially if this could get you out of having to work in a BSL-3.
Well, that's what I was going to say too. And the other comment that I would make is that this, I think, is proof positive that no one is taking any of this lightly. They are thinking and thinking and thinking of how can I work with something and still not endanger the world. All right. So here's one way to do it. There have been many other ways, I'm sure, but this is a great start to getting people to think that scientists as a whole are very responsible for their work.
Well, this was published in December, and it has not been part of the discussion. The discussion has simply been, zero risk. And
Right.
I don't think that's fair. I think this is a reasonable solution. Mm-hmm. So I'd love to hear from the opponents of gain-of-function experiments what they think about this, and also from those people doing them. What are the negatives of doing this? I don't see any, but I might be missing something because I don't work in the field. It seems to us that there's a very strong positive in that this is a self-destruct mechanism, essentially.
Yes.
Right?
Yeah, I could see two potential negatives right off the bat. One is this is an extra thing that you got to do.
Yeah.
And the other is that it could affect the biology of the virus in some way that you don't know. But that's why I would specify this is something you would do if you were kind of looking around more generally and doing an initial series of experiments, and then you could take this out and use the native virus for
a restricted number of experiments rather than doing lots and lots of experiments.
Yeah,
you could do a restricted number. And I think the public can understand this concept of a self-destruct mechanism. It can be presented in such a way to increase the safety of these experiments. And, all the hollering about dangers, I think this really negates a lot of it, right? So why are we ignoring it? In fact, I spoke to someone who's involved with this whole debate last week, and he said, I don't know, I've been suggesting this for a long time. Why aren't people doing this? So
which is not to say that the Kawaoka and Fouché work, as we discussed before, that was not done in any dangerous way. This is not something that needed to be added to that.
No.
But as I said, especially if this would allow you to work with a virus in BSL-2 that would otherwise have to be in BSL-3, this could be a huge boon. It could make things a whole lot easier.
Well, this is really to address the discussion, which, as Ryan said, is not going away.
Right.
So despite the fact that NIH has put into place additional requirements to do these kinds of experiments, the discussion continues, as we talked about last week. This is a way to deal with that.
Yep.
So, if you're really worried, put this miR192 sequence in and do your experiments.
Yeah. And Rich, you had a comment here in the
because we talked a lot last week about how there is already a policy in place, but we weren't specific about that. And so I just wanted to, we can put this in the show notes, I just wanted to, for the record, give the links to those. At the NIH, these matters are considered out of the NIH Office of Science Policy, and they have a web page that you can go to that has lots of resources on it, including links specifically for biodefense and biosecurity and biosafety. And if you dig around in there, you can find the policy that governs all of this, which is the NIH guidelines for research involving recombinant or synthetic nucleic acid molecules. And this originates back in the 70s, subsequent to what we've referred to as the Asilomar Conference, which was a gathering of scientists to consider the pros and cons and potential safety issues surrounding recombinant DNA research. And that's when this whole process was set up with institutional biosafety committees at the one end of a chain, a sequence of offices that one can go through, leading all the way up to the highest levels of NIH, to review proposals for various types of research that might have safety issues and decide whether or not the research should go forward. And those guidelines have been modified over time to accommodate different types of research other than recombinant DNA, including, most recently, dual-use research of concern and gain-of-function experiments. And the most recent modification was November 2013, so that's fairly recently, following on all of the hubbub about gain-of-function research to try and incorporate that into the guidelines. And that's there for everybody to read, and we can link that in the show notes as well.
Right. So the regulations are already
in place.
Yeah.
Yes.
Yep. They are. Okay. Now we have next a paper that was actually on last week's agenda, but we didn't get to. This was actually a paper suggested by Dr. Despommier.
Isn't that amazing?
No, it is not amazing because you read science religiously.
I do.
Well, especially now that he has glasses.
That's right. Well, they haven't come yet, though, Alan, so I can't actually say that I took advantage of that.
But
okay.
You know, Dickson, you are a traditionalist. You have a subscription to the paper issue of Science.
I do.
And you rip out articles, and Monday morning you put them on my desk.
I do.
Right?
I do.
And I have a whole bunch of them here.
Yes, you do, which you've barely read.
Dickson, that is really nasty comment that you make there. I love science.
I know you do.
I may read them. I may not understand them, but I do read them.
No, that's not true. You read them.
This one is called Lassa virus entry requires a trigger-induced receptor switch.
Indeed.
It was published in Science in June of this year. And the authors are J. Robin, Herbert, Kuhn, Racaniello, So, Stubbs, Jansen, Dom, Saftig, Whalen, Dye, and Brummelkamp.
You did that very well.
Yes.
It's not that good. It's okay. Anyway, Lassa virus. We haven't talked about Lassa since like episode 2, Dickson.
Yeah, that's true.
Lassa fever, which we recognized in the 1960s and had a huge story part right here at Columbia, right?
That's right. Correct.
And we talked about that. So we'll refer readers to those early podcasts where the sound quality was not so good, and you and I were not polished at all.
Our voices sounded more youthful.
Yes, we were much younger.
And the other thing about it is that it was the original virus outbreak which triggered the synthesis, if you will, of the BSL facility. And they created one at CDC just for the purpose of working on this virus.
It was funny. One of the nurses who was infected in Nigeria,
Nurse Pinio,
they just flew her back here on a plane sitting in the passenger compartment.
Without knowing what she had.
Yeah, of course.
But with knowing that about 5 other people around her had just died.
In Africa.
That's right.
Yeah,
Lassa,
funny.
Nigeria.
Things change.
So this is a virus that's changed lots of things.
It has changed lots of things. Yeah,
so we're gonna avoid those, please.
Oh,
why?
Sorry.
Oh, you of the title mania today, please.
Yeah,
that's right.
Interesting comment made here by Rich Condit.
So I looked up. I have to look up all this stuff. So this is it's an arenavirus.
Yes.
Which is ambisense, right? It's a two-part, mostly negative. Well, it packages what you could call the negative sense of the genome in 2 pieces, single-stranded RNA. But in fact, it's ambisense, so there's coding sequences on both the plus and the minus sense of the genome.
Yep.
Crazy.
Yep.
And this thing, Lassa fever, it's a classic zoonosis.
Yes.
The virus is equilibrated with a rodent, and so the rodent carries it around pretty much persistently, and it doesn't cause the rodent any problems. It reminds me very much of hantavirus, a bunyavirus, closely related.
Indeed.
But this can be passed to humans via aerosolized rodent urine or feces, and infect people and cause Lassa fever. It turns out to be about a 1% mortality rate, as I calculated, but there's a huge number of infections every year. There's like 500,000 infections, and so you got 5,000 deaths, and this is going on. Yeah, and this is going on all the time.
That's right.
And the interesting thing is, it's kind of like, it's almost ho-hum. We're used to this, so it doesn't get the press that Ebola does.
That's true.
When Ebola breaks out, it causes many fewer deaths, but it's big news. Lassa fever's going on all the time. It's kind of like traffic accidents, right? 40,000 people a year die on the highways in the U.S., so that's not news.
Or the pneumonia deaths.
40,000 people. How many people die of H5N1 every year?
About H5N1.
Yeah, right. 2.
I think we should focus on traffic accidents, but we don't want to interfere with the economy, right?
Of course not.
Yeah. So the reservoir is Mastomus natalensis, or Mastomus.
The multi-mammate rat.
And in Hamilton, Montana, they are maintaining a colony of these mice.
Multi-mammate rats.
Isn't that interesting?
The rats. The
natural Mastomus is a multi-mammoth rat.
That's
but it is a rodent.
Yes.
Most common rodent in equatorial Africa and eaten as a delicacy in some areas.
Indeed.
Oh. So I didn't look into this carefully enough. Is there human-to-human transmission of this, or are these all primary zoonotic infections? Do you know?
It's sort of like Ebola. You need to have close contact, healthcare worker, families, that sort of thing.
Yeah.
Certainly no aerosol transmission. And as we will see in the paper, we will understand why that is, right, Dickson?
Yes, we will.
So this is a cool virus, and I must say that this is the virus that inspired me to become a virologist.
Oh, wow.
So I bow down to Lassa. Okay.
And to John Frame. Thank you, John.
Thank you, Lassa and John Frame.
That's right.
Thank you for framing me. Okay, this is a cool virus. Now, this paper is awesome. So it has been known for a long time, many, many years, that there's a self-surface receptor for Lassa virus is called alpha-dystroglan. It is a glycoprotein on the cell surface.
Right.
And I remember that paper had Michael Oldstone on it.
Dystroglycan.
What did I say?
Dystroglan.
I said dystroglan?
You left out the gly.
Sorry.
That's right.
Dystroglycan. Mike Oldstone on the paper.
A deletion. You inserted.
You had a deletion.
It wasn't framed.
Deleted a syllable.
Cell paper.
One verbal codon.
Some kind of subliminal deletion.
That's right.
Alpha-dystroglycan. It actually flows better, dystroglan, but that's a different protein, isn't it?
That's right. Or an organ.
It's a receptor, and it's also another important thing to note here. The virus infects many different species of cells, but not chicken.
Correct.
Not chicken cells.
Nope.
And in this paper, they actually don't use the virus itself. They use pseudotypes, which means they take vesicular stomatitis virus and they give it the Lassa virus glycoprotein.
Look at that.
That way they can work at BSL-2 conditions. So that's another way to get around issues, right?
Yeah.
If you want to study entry, which is what they're doing here. And in fact, the Ebola virus entry paper that we did a long time ago from Kartik Chandran, where they identified Niemann-Pick C1 protein as they use pseudotype viruses also. You can do that.
Yep.
So they want to know why there's a defect in entry of Lassa virus into chicken cells. And that is also the fact with a pseudotyped VSV with the Lassa glycoprotein, it doesn't get into chicken cells. But chicken cells have alpha-dystroglan.
They do.
Dystroglycan. I'm going to do that for the entire show.
It's okay.
Alpha-DG.
Alpha-DG, that's good.
That's it.
There you go.
So they want to know what else is involved in entry. So they're looking for other entry mediators.
Indeed.
Okay? And so they do a haploid screen. And we've talked about this before. It was a long time ago. There is a cell line that happens to be haploid. Just like you and I were before, we were
That's
born.
right. We were all at one time haploid. That's right.
That was Rich Condit's original when you were haploid.
It was a good one.
Where were you when you were haploid, Dickson? Were you in New Orleans?
He doesn't remember.
No, I was in the zona pellicula.
So the idea here is that you want to take. Let's say you infect cells with a retrovirus, and they will insert their genome randomly, and so you could use it sort of as a mutagen to identify genes that are important for whatever you're looking at. But if you have 2 copies of every gene, it makes it hard.
Right.
Right? Because you're never going to knock out both.
Yeah.
Right.
So, but amazingly, someone found this haploid cell line years ago. And it's called HAP1.
Amazing.
So now you can basically infect them and make a library of deletion or knockout mutations. And then if you have a selection, you can apply it, right? So they start actually with cells that don't have alpha DG.
Right.
See, I remembered to call it alpha DG. and just... for their purposes, it turns out that the virus, the pseudotype virus, can infect these cells without alpha DG. And you may say, well, how does that work? And well, sometimes you get lucky. It turns out that it binds heparan sulfate and can get in. It's like an alternate entry pathway. So you take these mutagenized haploid cells lacking alpha DG, you infect them with the pseudotype virus, and then you say who survives. And of the survivors, what genes are knocked out. Right?
Right.
Right.
And then you make this cool table with circles of
Sounds like Huey, Louie, and Dewey just now.
Importantly, the retrovirus mutagen serves actually 2 purposes.
Right.
Not only does it mutate, but as it does so, you're introducing a provirus, so the mutant gene is marked with the provirus. So when you get the survivors out, you can actually sequence away from the provirus and identify the gene that's been knocked out in those survivors. So you not only get the mutants, but you identify what genes are mutant.
I love the way the visual information is presented.
Yeah.
It's easy to see, it's a visual thing, but it's still quantitative.
Yeah, so they do this
It's fantastic.
In bulk, and then they sequence, and they say what genes are most often associated with resistance to infection. And then, the more significant, the more times a gene is hit or scored in this assay, the bigger the circle.
Right.
So you have a lot of noise of little tiny points at the bottom, and then you get higher and higher in more significant areas, and you get genes. And they can organize them into pathways, like some are involved with sugar or Golgi or vesicle trafficking. And then we have our heparan sulfate involved genes, which makes sense. Got cosylation genes, and then genes in the lysosome. Okay.
Right.
You get a lot of information out of that. So basically they say there's a protein called LAMP1, which turned out to be very important. They got 127 hits for LAMP1. And also proteins involved in glycosylation and adding of sialic acid residues, in particular one gene called alpha-2,3 sialyltransferase, which is ST3-gal4. Okay? So LAMP1 and ST3-gal4. So if you knock out either one of those, the virus will not infect cells, just like if you knock out alpha DG, right?
Yep.
So alpha DG, LAMP1, and ST3-gal4 are really important. So what are they doing? That's what we're going to figure out in this paper. If you put human LAMP1 in chicken cells, the virus will infect the pseudotype virus. But if you put chicken LAMP1 in chicken cells, that doesn't work. So maybe there's some problem with LAMP1 in chicken cells. Okay? Now, what is LAMP1 doing? LAMP1 is a protein on the endosome inner membrane, and in fact, it is localized in acidic lysosomes. All right? And what they do is they produce the Lassa virus glycoprotein, and they show that it will actually bind not only to alpha-dystroglycan, but also to LAMP1, and LAMP1 it will bind to only at low pH. So we'll bind alpha DG at neutral pH, and we'll bind LAMP1 at low pH only. And in fact, if you bind the glycoprotein to alpha DG, it will then bind LAMP1 if you lower the pH. So binding to one doesn't prevent the second one. So you see there's something arising here. There's an idea of what's happening as the virus is coming in. Mm-hmm. They put GFP into their pseudotype virus so they could look and see what's going on during infection. So if you infect cells that lack LAMP1 or that ST3-gal4 protein, the virus gets hung up in vesicles in the cell. Mm-hmm. So their model is that you need LAMP1 for the virus to get out of the endosome, basically. Right? You got that?
Yep.
The way this works...
So the virus is getting pulled into the endosome. The endosome is going to fuse with a lysosome and get acidified.
Right.
And if you don't have LAMP1 there, it's trapped in the endosome. LAMP1 seems to be crucial for getting it out of the endosome at low pH.
That's right. And what happens at low pH is that the viral glycoprotein is actually has two, and Rich has put in a picture here, it has two chains, and there's a G1 and a G2 subunit. The G1 is not covalently attached to G2, and at low pH it dissociates, and that allows then the glycoprotein to bind to LAMP1. Mm-hmm. Okay, so the model is the virus binds alpha DG at the cell surface, as Alan just said, gets into the cell, and then upon acidification it binds LAMP1, which allows fusion. So they produce a form of LAMP1 that goes to the cell surface. This is a cool experiment. All right, they can make a change in the sequence.
Right.
It doesn't stay in endosomes anymore. It goes to the cell surface. And if you lower the pH of those cells, guess what happens?
Let me think. The virus binds to the surface.
No, this is just the LAMP1 mutant protein expressed on the cell surface. And I'm not supposed to say expressed, Jane, I'm sorry. If you take those cells and then lower the pH of the medium, what do you think happens?
There's a dead silence.
All right, the cells fuse, man. They fuse.
Right.
So
I said bind. I thought I said that.
You said the virus, but that's okay, Dickson.
Oh, the cells bind. Okay, so
now they take these same cells. They got LAMP1 on the surface, right?
Right.
You add the pseudotyped VSV with the Lassa glycoprotein. Mm-hmm. At neutral pH, do you get infection?
No.
If you lower the pH,
yes,
you lower the extracellular pH. You now allow fusion because that's what you need, low pH, for the virus to engage LAMP1 and to fuse.
So you've essentially turned the outside of the cell into an endosome.
Exactly.
Sure.
It's very cool.
Sure.
So that's why LAMP1 is important, because it's a second entry receptor. It's in the endosome, and this is very much like the Niemann-Pick C1 protein that Kartik Chandran identified for Ebola virus. It's an endosomal protein needed for fusion. We talked about that in TWiV 166. Boy, that seems like a long time. Doesn't it, though?
It is.
Yeah,
it was a long
time ago. It was a couple of years.
That's right.
Holy cow.
I know.
We got like a legacy here, don't we?
We do.
All right, now they want to know what this ST3-gal4 is doing, right? Yeah. So LAMP1 we know is a fusion receptor in the endosome. What is ST3-gal4 doing? So they did another haploid screen, this time starting with cells lacking ST3-gal4. Okay?
Right.
Got that? Because you want to know, if you take that away, is there something else that can work to get the virus in? But they didn't pick up LAMP1 under these conditions. So LAMP1 wasn't picked up in this haploid screen. Only when ST3-gal4 is present does LAMP1 show up as something that you need for virus entry.
Implying that there's a relationship between these two.
Yeah, so they say, all right, these two must be related. What is it? So it turns out that LAMP1 has a couple of domains, one of which is highly glycosylated. It's got how many different glycosylation sites? So they just basically change them one by one and ask which one is important for virus entry. And it turns out that there's an asparagine at amino acid 76 that you absolutely need to get arenavirus glycoprotein binding and entry.
That is amazing.
Okay?
And guess what?
Yeah, guess what, Dickson?
Chickens don't have that.
Yeah.
Right.
That's right. But if you put it in the chicken, LAMP1.
Yeah, it turns into a person.
And if you
yes,
if you put the chicken 76, well, if you take out the 76 from human, then it doesnn't work anymore.
Right.
So basically, they believe that the sialotransferase ST3-gal is sialylating this asparagine 76
Neat.
on LAMP1, making it a fusion receptor for
Bingo.
the arenavirus glycoprotein. And that's why you need both LAMP1 and ST3 Gal4. It's really cool. It's very cool. They knock out LAMP1 in mice, and they show that the virus doesn't replicate. It's cleared. So you do need LAMP1 in the natural rodent reservoir.
In all honesty, by the way, when I cut this article out and brought it into Vince, I had no idea what it meant.
You just like that it was Lassa virus?
Yeah, no, it was a good article on Lassa virus.
Well, you hit it, man.
And it was molecular. And then I had him explain it to me. And he sat here without notes and did this show for me before the show started, just so I would be able to follow what's being said during the show.
See,
and I think that's
tell me fantastic. I don't treat you well, Dickson.
No. I never said that. In fact, I'm now saying that. You treat me really well. You treat me like a little baby.
So here's the cool thing in closing on this. So the ST3 Gal4 is an alpha-2,3 sialic acid addition enzyme, sialotransferase, right? And if you remember, if influenza viruses like avian influenza viruses like alpha-2,3 linked sialic acids, right?
That's right.
So they say here humans have largely alpha-2,6 in the upper tract and alpha-2,3 in the lower tract. Right. And they make this suggestion that maybe humans evolved to be that way, to be resistant to avian influenza viruses. I don't know if that's true or not, but it's a speculation. Yeah. And then they say, and that might make people resistant to aerosol transmission of Lassa virus, because in our tract we don't have alpha-2,3, which we know that LAMP1 needs to have in order for uptake.
Yeah.
So here's the experiment.
I'm sitting here waiting with bated breath. Yeah. Come on, tell us.
We're going to take people and change their sialic acids in the upper tract to alpha-2,3.
No.
You can't do
what could go wrong with that?
Right, exactly.
Now, so in all seriousness,
yes.
Okay.
Yes.
Would it be informative to passage or adapt Lassa in some way to make it recognize alpha-2,6 sialic acids?
Ooh.
I don't think so. I don't think we know that what would happen. It would be transmissible probably. So I would say ferrets have mostly alpha-2,6, right? This probably doesn't transmit among ferrets. You could probably select a variant that would, but I'm not sure that would tell you anything. Yeah. And it would probably be very dangerous for people, right? That would be BSL-4.
This
is not a respiratory virus.
No.
Right. I mean, you inhale the mouse feces or whatever.
So that must go down into your lower tract when you do
right.
that.
It gets
right.
And presumably people are getting this into their lower tract where it finds the right receptor. I don't know. If somebody wanted to try to make the case for why that experiment would be worthwhile, it might be worth hearing them out. But I don't initially see it.
Lassa and related viruses don't ordinarily cause pandemics.
Right.
So in terms of the larger biology of the situation, the experiment is not as relevant. I won't say irrelevant, but not as relevant as doing the same set of experiments with flu.
Sure. Also, you could do a similar experiment with a pseudotype virus. Right, since it's an entry question.
Right.
Absolutely,
So you could answer that question without any of these risks.
Yep.
Yeah. Although, with H5N1, it's not just entry changes that are important for transmission, right? So
right.
You may get it to attach and enter in the upper tract of ferrets, but it may not still transmit.
So I can add something to this story a little bit from an ecological standpoint, and that is that there are seasons for this transmission. And in drought years in West Africa, which this may be, the crop production is low, and the natalensis is a wild rat. This is not peridomestic. These rats live in the wild, outside the farms. And they have enough to eat when times are good, but when times are bad, and the food supply outside is dwindling, the stored grain from last year's crop is a food source, not only for the people, but also for the rats. And the rats then invade the settlements, and that's how people come in contact with natalensis. So you were saying before, Rich, that it reminded you a little bit of hantavirus because
sounds like it,
it's the urine of the mice that cause the respiratory infection. I knew someone who was in the outbreak that defined hanta in the United States, the Four Corners disease, as they used to call it.
Yep.
And he became a graduate student here, basically. And he told me that while he was working for the CDC, they did surveys of houses and throughout the Navajo nation, and discovered, to their amazement, that if there were fewer than 3 mice per house, there were no cases of hanta. But if there were 4 or more mice found within a household, you could pinpoint a case. And that was remarkable, considering the threshold for transmission. And by the way, the hantavirus behaves similarly in terms of the mice. When there's a bumper crop of mice the next year, you're going to expect an outbreak of hanta someplace, right?
Right.
So I'm just wondering how many Natalensis rats you need per hut or per boma or per living space in the rural districts where this is a problem.
Or per grain storage
Or
silo.
Yeah, exactly right. Well, that's not how it works out there, though.
Right,
Every
right.
house ends up with their own grain supply. So I'm just wondering whether there's a correlation with loss of virus transmission for number of rats or number of rat hits, perhaps.
Somebody's probably done the math.
Maybe. And it would be very
I
interesting
guess,
to know that, because it would also tell you what the threshold is for encountering the virus in the environment. Right?
I was really, as I said before, really astonished to learn how many cases of this
it's a
annually
lot.
we're dealing with.
A
It's
lot of cases. In the beginning, by the way, when Nurse Pinio got sick and she was sent back here, it was estimated that the case mortality rate, number of people sick versus number of people who died of those people who were sick, was up around 80%. And then, of course, later on, they went back and looked, and they found it was much, much lower than that. And, in fact, John Frame, who was involved in this original outbreak, was hired by the Army to go out and collect convalescent serum from people who were tested first for Lassa, found to be positive, but had no symptoms or anything else. Those people were asked to donate their sera to this bank of immune serum kept at Fort Detrick, while we were exploring the biological aspects of Lassa fever.
So that was TWiV9.
TWiV number
Fever,
9.
the discovery of Lassa
TWiV
virus.
number
I got to read this. Listen. Vincent and Dick. I used to call you Dick
You
back
did,
then,
you did.
until you said, I don't like that.
No, that's okay.
And I don't like Vince. Recall the discovery of Lassa virus in Africa in 1969, a non-fictional account of the story Fever, written by John G. Fuller and published in 1974, inspired Vincent to become a virologist. Part of the story took place at Columbia Presbyterian Hospital, where both Vincent and Dick are employed. Dick remembers many of the key players in this medical drama.
Yeah, he doesn't anymore, but he used to.
I guess I'm going to have to read that book, huh?
Oh, yes, you should. There's a link to a used copy over at Amazon in our show notes, yeah.
And by the way, they made a docudrama over that also.
It's a really good book. It's really well done.
Yeah.
And that was really the first emerging infection, right?
That's
sort of alerted us to the fact that
That's correct.
Yeah.
viruses do zoonotic infections, right?
Exactly.
Okay. You want to do some email? Oh,
Yeah.
Nobody wants to.
Well, I just
No, let's do
before we leave this paper, I just wanted to mention
Yep.
You were commenting on supplementary figures. This one, there are 22 supplementary figures and 4
Lordy.
tables.
Lordy.
Yeah.
So for those of you reading along at home, you might want to.
So
look
you
it
know,
up.
I think this paper is loses something by being in science. It is so compact. Often one sentence will be the conclusion of 5 different figures.
Yeah.
I think it's unfair.
Well, it's an enormous body of work.
It's wonderful. It's a wonderful, enormous body of work, and I think
Yeah.
it's getting short shrift by being here, unfortunately.
Yeah, the introduction is what, 1 paragraph or something,
Yeah.
and then bang, you're into the science, and yeah, every sentence is a year of graduate student time.
Yeah,
or something.
I'm glad you brought this up because I found it really dense and really difficult to read as a if you're a graduate student or something like that, it would be nearly impossible to read.
I'm feeling better now, as you guys are saying.
But I think this is just. I understand they want to put this in a high-profile journal. I understand completely. But why does science have to do this to wonderful data? Why do they have to make it indigestible? Mm-hmm. They've made some editorial decision some time ago that to be profitable or to break even or whatever, they have to do this. But it is not fair to science at all. I would have loved to have read a nice, detailed with every figure there in the text.
Well, you see, if there weren't people like Dickson still receiving the print issue, they could just go all online and they'd have unlimited space.
Oh, it's your fault.
Yeah. Oh, yeah. Sure. It's my fault. I have to have something to read over breakfast. This is my science.
You have an iPad, Dickson. You could just fire it up and...
I'm old school.
You just keep telling yourself that, and eventually you'll really be old school. All right, let's do some email. Yeah. Alan Dove, can you take the first one, please?
Sure. Neil writes, Dear TWiV hosts, I'm behind in listening to episodes of TWiV, but I took note of the discussion during TWiV 280 of the commentary by Ferris Jobber on the concept of life. As Vincent may recall from early last year, I engaged in a brief correspondence with him regarding the living, non-living status of viruses due to the online poll on the TWiV site. This correspondence prompted me to write about the issue for the Evolution and Medicine Review, for which I contribute posts on a roughly monthly basis. And he gives a link to that article. By the way, I'm utterly unconvinced by Jobber's argument. I'm confident that if someone transformed one of his loved ones from meeting the putatively culturally determined criteria for alive into meeting the putatively culturally determined criteria for not alive, he would recognize the change as of real significance, independent of cultural factors.
It's great.
Of course, in medical practice, defining this boundary precisely, however vexing, is of great practical significance. I also took note of Vincent's mention of a disagreement he had with Carl Zimmer. Last year, I published a brief essay, he's linked to that takes note of a claim by Carl Zimmer during an interview on Fresh Air with a guest host that bacteria were more similar to human cells than were viruses.
Very gross.
I exploit this questionable assertion to explore the complexities of the idea of similarity. The arguments I offer may also be relevant to the issue of defining life. Not directly related to the preceding, I have a question. Many years ago, I heard Eckhard Wimmer give a lecture about his studies on polioviruses. As best I can recall, he mentioned that a portion of the poliovirus genome of one serotype encoding one of the gene products was closer in sequence to a comparable region of a non-poliovirus picornavirus than to the corresponding sequences of polioviruses of the other two serotypes. This relationship made me wonder if what we call polioviruses are just picornaviruses that happen to have evolved neurovirulence. If so, and if public health officials eventually succeed in eradicating current polioviruses, how likely do you think it would be for some other picornavirus to evolve neurovirulence and recreate a polio-like illness? Finally, did TWiV take note of the 40th anniversary this past April 19th of the discovery of MHC restriction of T-cell recognition, generally attributed to Peter Doherty? Disclosure, my PhD thesis advisor, and Rolf Zinkernagel for their work on the murine CTL response to lymphocytic choriomeningitis virus. Although there were some similar earlier findings with what we later termed CD4 positive T cells, Doherty and Zinkernagel were the ones who most clearly interpreted the phenomenon and related it to explaining the extensive polymorphism of MHC genes. This advance had an enormous impact on immunology and especially viral immunology.
Well, I just met Peter Doherty in Melbourne when I was there. Oh. I did an interview for him, which will be in the next edition of our virology textbook.
Right.
We didn't notice that anniversary at all, did we?
No.
Nope.
No,
so there you go. And that's pretty cool.
We noticed it now.
We did now. Thank you, Neil. Neil is at Case Western, by the way. All right. Well, this is an interesting question about polio. Well, poliovirus, the way you classify things is just really rapidly evolving, because now if you look at all the nucleotide sequences, polio and rhinoviruses are very, very, very related. And, they're biologically quite different. So I'm not sure what you say that polios are just picornas that happen to evolve neurovirulence. I'm not sure that is right. That's probably too simple, because there are other picornas that are neurovirulent as well.
Right. That's what I was going to ask.
Now, the question of whether something else will emerge after eradication is something we all talk about, and we don't know, because there are neurovirulent polioviruses out there, and many of the guts of humans, they are replicating and being shed. In fact, when you take the Sabin vaccine by mouth, and it replicates in your gut, it recombines with enteroviruses that are already there. And so the viruses that you shed are all pretty much recombinant with those enteric enteroviruses. Very interesting. I don't know. Yeah, it could be one will emerge. Who knows? Who knows what are... I mean, polio emerged during a time of poor sanitation where fecal oral contamination was very easy. And it may be that those conditions don't exist globally in a sufficient level, right?
Yeah, I'll ask a question here, just because we're on the topic of poliovirus. When is the first documented case of poliovirus in history?
How do you mean by
Noted.
documented?
Something that you could identify as an outbreak of polio rather than something else.
Well, so there have been sporadic reports over the years, starting from the Egyptian stele, right? But it looks like polio, but who knows, because we didn't isolate virus. And you have all kinds of reports over the years until the end of the 1800s where there was an outbreak in Sweden, which was clearly an infectious process, and there was paralysis involved. And then in 1906, the virus first isolated from a human. So probably at the beginning of the 1900s is the definitive answer to that.
But your assumption is that the virus has been around a long time.
I think it's been around for thousands of years in people, yes.
Right.
And I just don't know. I think it's a really interesting question whether once we get rid of it, something else will evolve. Who knows, right?
Because I think smallpox, there's clear records. You go back to the pictograms and that sort of
has
things.
such a clear picture, right?
It does. But cholera, for instance, on the other hand, which is obviously not a viral disease, but rather mitigated by a virus inside the bacteria, right, didn't occur until 1817. They know exactly when it occurred because it has such characteristic symptoms that you would have known about that before from the literature that existed prior to that time. So very interesting.
By the way,
now I just wanted to skip back to this classification issue. Do all three polio serotypes cluster tighter to each other than they do to any other picornaviruses, or is this correct that one serotype might be more closely related to something else?
No, if you take the whole genome, then they cluster.
He's talking about a portion of the genome.
So
he's talking about a portion.
So if you
of
just
it.
take a portion, then that's probably true. But if you look at the whole genome, they clearly cluster with themselves. They're very close to the rhinoviruses.
Okay, that's
why they're all enteroviruses as well.
But a segment of it may have come from someplace else, and that's not terribly surprising.
Not at all. Not at all. No. He makes some interesting comments about. I would suggest that listeners go check out his article on evolution in review, and that's actually a discussion of our discussion here on TWiV about whether viruses are living or not, and also his commentary on Carl Zimmer, which is linked, is very interesting as well. Clearly, he has thought about these things extensively. They're very well written. All right, Rich, can you take the next one, please?
Sure. Chris writes, "Dear wonderful TWiV hosts, I loved the discussion of rescuing biomedical research, paper from TWiV 282, and I'm curious to hear your opinions on some thoughts. The paper notes the surplus of excellent biomedical researchers being churned out of PhD programs. Sorry. Little slip there.
Whoops.
And the hyper-competitive system it has produced that is not healthy for the field of biomedical research. I came across a different paper the other day that reviews a collaboration between Tufts Medical School and the Boston Public Schools to bring new biology curricula to the public high schools, and it gives a link to that. Their overarching goal was to increase scientific literacy and influence health-related decisions. The new curriculum focuses on what they consider the great diseases, infectious diseases, neurological disorders, metabolic disease, and cancer, and is structured on the premise that by providing content more relevant to students' lives, they will both increase student engagement and increase student health literacy. As a public high school employee myself, I can attest to how uninspired some of the current science curricula is and the disinterest it generates. So this is addressing a real problem. It is also amazing how human biology and disease is such a minor part of high school science, at least in Washington State, even though it is a massively relevant topic. I'm curious if any of you have seen studies looking for a correlation between the knowledge of human biology and healthy lifestyle choices. This paper never states it explicitly, but I would imagine that the correlation is not zero, which could provide an inexpensive way of improving public health. Given the successes described in the paper, I'm curious if you see any potential for the expansion of biomedical science and scientists into high schools. In this case, all of the participating high school teachers were paired with a biomedical expert who they were trained by and could contact to ensure they were presenting correct material. I'm sure there are a myriad of other similar collaborations, other ways similar collaborations could play out as well, maybe with even more direct involvement in the school by biomedical experts. As a biomedical expert hopeful, I'm currently applying to medical school, with a passion for teaching, I imagine there are at least some similarly minded people in the biomedical sciences. Seems to me like this kind of model has the potential to be a big win for the education system, public health, science communication, and could ease at least some of the burden stressing the current biomedical research system. Thoughts? Weather here in Seattle is currently 16 degrees C with, surprise, a chance of rain. Thank you for all you do. You are educating and inspiring a generation of scientists like myself. Mm-hmm. So I know that the link to this program she describes is, Chris describes is great.
Yeah, it's cool.
I know that I'm just thinking, I can't describe it exactly, but I know that there's an ongoing program here at the University of Florida where high school teachers are brought into laboratories during the summer to gain experience in a laboratory that they can, basically increase their understanding of research and their ability to address relevant problems back in their classrooms. My guess is that those kinds of problems, you say there's one at Columbia.
Yeah.
There is a big one.
Those kind of programs are probably not uncommon.
I think they're good. I mean, I like this idea of having, medical school faculty participate in the teaching. In this particular example, which is really nicely described in this article, they got financial support to do that. And that is the key, because the people in medical schools typically do things to pay for their salaries, especially the clinical people, right?
Yeah.
And others raise grant money. So it's always going to be a trade-off, right?
Yes.
Between getting people to spend their time doing something. I think this is a great idea.
Agreed.
But I just think practically it's hard. But maybe training another generation of PhD students who do this is a useful thing.
Well, there's another issue that we should discuss here, too. And I've had huge numbers of encounters with these teachers because I was part of the program for 12 consecutive years and had one teacher in my lab when I still had a lab, and then continued to remain in touch with this subject. And I offered myself up as a visitor to their classroom situation, and a lot of them took me up on it, and I was really quite pleased with the response of the students, et cetera, et cetera. But I've also had experiences where high school biology teachers, particularly, and not in school systems like New York, teach via a set program. And that set program says, we're going to do the lung today and the heart tomorrow and the brain next week and that sort of thing. Or we'll take an evolutionary approach and we'll teach that. But what they don't like, and I can tell you this from personal experience of just talking with a few people outside of this arena, is that if someone of expertise comes in and sits in class and offers themselves as a teacher and teaches some aspect of biology that they're currently encountering in their studies, and then leaves, the teacher that's left behind often feels not inadequate because they are high school biology teachers after all, but rather the students expect more from that teacher after that because the information given by the person that's visiting is at a higher level. Always. And so it turns out that you don't get invited back a lot to those schools. And it's not a funny thing, though, because I I love teaching and I love being in those situations, but I understand what it would be like to be a high school biology teacher, to have someone come in and say, and now we're going to hear from an expert on such and so, and then they leave and we go back to page 23, and then and it's really a downer for the class afterwards. And so I don't know how you get around that without having two people in the same classroom all the time.
There are huge barriers to stuff like this.
Yeah, I know.
I have had some fairly direct contact with it. Well, my dad was a high school biology teacher for 40 years.
Oh, wow.
But I also did some volunteering when I was in graduate school in the New York City public schools teaching speech and debate. And I came in as an outsider and, provided stuff that they weren't easily able to get from their regular teachers.
Yeah, exactly.
And had kind of the same experience you had, but it was part of a more structured setup, so that they almost had to keep inviting me back, which helped a lot. But that was orchestrated by an outside nonprofit organization.
Got it.
Which had funding, they had donors, they had so there's the cash problem, there's the staffing problem.
That's right.
And there's there's an incredible institutional inertia here. If you pick up a high school biology textbook, I don't know if you've had this experience, but it's depressing.
It is.
To look at what's being presented and the way it's being presented, and it's often incredibly convoluted.
And how out of date it is.
and out of date, but even if you get one that just came out, of course, the schools aren't going to be able to buy it for another 15 years because they don't have the budget to recycle their textbooks every year.
Yep.
But you look at it and there's all this material in there that is in many cases structured to be lists of facts that are easy to test.
Right.
Well, that's what it's all about, right?
And the reason for that is because we've structured our entire educational system around measuring test results.
Yeah. That's right.
And this has only gotten worse in recent years. And on top of that, the textbook publishers are, people often say, well, it's the fault of the textbook publishers. Well, the textbook publishers are in business. And they need to sell books. And the way you do that is to target your books to the biggest textbook markets in the United States.
Right.
Which are Texas and California.
That's true.
So you build your textbook around the curricula and especially around the tests of those two states, and everybody else can just suck it up because that's the book that's available.
Mm-hmm. Mm-hmm.
I'm just scratching the surface here. I could go on and on, but
yes, you're absolutely right. Because my middle-aged son, he comes home and he says, they're teaching us to do well on a test. I
know.
Yeah.
Not what's interesting.
That's right.
But this is sort of triggered an idea, which is that I think his idea of teaching human biology, why don't we start from something that is real for kids, human biology, teach them that, and then when you have to teach them something important to understand it, then branch off and teach it, rather than starting with, the way biology is taught, step by step, trying to give them everything they need, but they don't know why it's relevant, right?
Mm-hmm. True. The moment you get to human reproduction, however, this is a highly sensitive issue.
Yeah, I understand.
Highly sensitive. The other thing that saves the day for some schools is AP Biology, where you've got advanced courses. You can use an advanced system of teaching. You don't mind bringing in experts for various subjects.
Yeah, but Dickson, all they do is go faster in some of those courses.
I do know that,
well,
too.
And the other problem with AP is it just... it says, okay, we're going to admit that the system sucks, so we're at least going to try and save the very best.
No, you're right.
We're going to save the top 10% of students who were probably going to do fine anyway.
Yes.
They're going on to college to heck with everybody else.
The only systems that work at that level is chemistry and physics, I think.
Which, unfortunately, we teach science backward in this country.
Yeah.
And we start off with biology because it's perceived as easier, which I don't know who got that idea. But
well, the way it's taught is easier.
And then the only people who get chemistry and physics are the advanced students. How are you supposed to understand biology if you don't get chemistry? And how do you get chemistry if you don't get physics?
Got it all right.
This is all part of the same Gordian knot. And by the way, the idea of scientists, professional scientists, academic researchers going and helping out. This is not new.
No.
In fact, the NIH has, or at least had, an organized program doing exactly this. And I don't know, Vincent, do you remember Dave Vanier, who was in my class?
I do.
He was running it.
So they funded. The NIH funded this particular project described in this article. Yeah,
and for a number of years they've been doing stuff, and Dave's been organizing this stuff where they build lesson plans that fit the required national and state curriculum. We don't really have much of a national curriculum because we're 50 kingdoms of education.
There you go.
But they build these modules that you can just plug into your science curriculum, and it's built around things like disease biology.
Yeah, I think
should
that's
be more
great.
relevant
That's
to
great.
students.
I'm going to contact Dave. He's still doing this, I presume?
I think he is. I just Googled him and I found a press release from 2011, but you could...
I'd love to have him on and have him talk about this.
That'd be great.
The program here was funded by NSF. So it was more of a general science.
Which one? Out here
at
Columbia.
The one at the medical center. So even though these were science teachers, they weren't called biology teachers, they're just science teachers. They could be physics, chemistry, etc. They all came to the health sciences centers to get an enrichment and working in the lab.
That's great. That works.
But some of them ended up working with atomic force field microscopes and others with X-ray machines and that sort of thing. So it did apply to their science as well. And they had an open mind. Those teachers were all younger. They had all had an excellent educational background to begin with. Many of them had master's degrees. Some were going for their PhD at that point. So,
and 2/3 of them are probably working in something besides teaching now.
Exactly.
Yeah.
I think that first year high school, you should take virology and then spend the next 3 years explaining what it meant.
You can't finish virology without cell biology first.
No, we can't. This.
I wish I had
a student. I had a
Well,
freshman.
if you start with physics in elementary school, then you could be up to that point.
I just think teaching them something that is life relevant
Yeah.
would help. So I had a freshman take my virology course this year, and she's not supposed to because she's supposed to have biology as a prereq. But she came up to me in the last day. She aced the course. She was great, right? She said, now I'll get biology next year. And that's true because virology is one of those, all-encompassing subjects that I think teaches you what biology and chemistry mean.
It's the monkey wrench in the normal biology.
Dickson, can you take the next one?
I sure can. You give me all the short ones, but that's fine because I'm limited in my ability to.
No, that was just the luck of the draw, Dickson.
Catherine writes, hello, esteemed TWiV team. I heard about this on the Nature podcast, and she gives a reference. How is this going to affect virology research given the common usage of the HeLa cell line? Will it only be relevant for researchers working on drug development? Is there any reason to think that topics such as viral attachment or receptor identification, two things I can think of off the top of my head, would be affected by the sex of the cells? Thanks for your thoughts on this. Love the podcast, and I look forward to Sundays so I can get my TWiV fix.
Oh, yeah. She links to an article which is NIH to balance sex in cell and animal studies.
Right.
The director says he's unveiled policies to ensure that preclinical research funded by the NIH considers females and males.
Yeah.
So preclinical is typically, leading up to a human clinical trial.
Right.
And so at my level studying, so we use HeLa cells, which are derived from a female, of course. I would think that basic viral replication, receptors and entry would be the same in male and female cells. However, I'm sure that patterns of gene expression are slightly different. So if you're, looking at global gene changes after virus infection, maybe you'd want to consider looking in male and female cells. The problem is that how are you going to match them?
Is by the way, the
right?
paper we just discussed with haploid cells, is that a male or a female cell line?
I don't know.
That's a good question. I don't know what the sex chromosome content of those cells is.
I don't know.
Or does it have a normal complement of chromosomes anyway?
I think it does. It's just got, one end. But
this is a great... I meant to ask another question here, too. Mm-hmm. Can you ever, in that cell line, eliminate chromosomes and still have a cell?
I don't know. You can eliminate single genes, of course.
No, but it's either male or female. So they've got sex cells. So they've got sex chromosomes. So does the sex chromosome contribute to the normal running of a cell?
I don't know, Dickson.
You don't know?
This is This Week in Virology.
You
I'm
don't-
sorry.
I thought they taught you everything about cell biology.
I forgot.
I'm sorry. I'm just playing devil's advocate.
Anyone know? Alan, do you know?
I'm sorry. I was trying to figure out if the sex of Hap1 cells.
Okay. The question is, do you need the X chromosome or the Y chromosome? Which one did you
Or
say?
both.
The people are working
You don't
on
need
that.
both, obviously, but
to do what?
to do anything.
To
To
live.
just
Could you
run the
eliminate
show.
either one of those
the-
chromosomes and still have a competent cell?
The Y chromosome contains very little.
We all
Actually,
knew
that's-
that.
I mean, it's a very small chromosome that.
But it's got genes on it that are important.
It does
though.
have genes. It's got the SRY gene, which determines, I think, most of maleness, and then a couple of other things. I'm not up on the latest on this, but I remember sitting through a talk a number of years ago about this. That it's there's not a whole lot in there.
Actually, there's a great diagram of the Y chromosome that shows the genes that are important, including channel surfing.
Right.
Channel surfing, that's good. I like it.
That's the SRF gene. The
That's right.
addiction to death and destruction movies.
Like it.
That's a T2 gene.
Like it.
The need to always be right. That one is called told you.
That's right.
So
And the lack of
there's,
ability to ask for directions.
It goes on.
There's a guy at MIT, his name is David Page, and he studies the Y chromosome. He just released a study this year showing that some of the genes on the Y chromosome are essential for male survival.
But that doesn't answer the question. In vitro, when you've got a cell line that's haploid, and if it's a male cell line, could you ablate the male chromosome and still have a normal cell?
I don't know the answer to that question.
There are a dozen genes on the Y that are expressed in cells and tissues throughout the body.
It's less than on a mitochondrion, for gosh sakes. This is sad to realize.
He said we're transitioning from the rotting Y theory. Which says the Y is going
How
away.
many are on the X chromosome?
Billions and billions.
Well, that explains everything then, doesn't it?
Each of the Y genes has a conserved counterpart on the X chromosome.
Yeah, we knew that, but how many on the X chromosome altogether?
You keep asking questions, Dickson.
Well, I want to know. I'm an inquiring
I can
mind.
tell you how many different kinds of viral genomes there are. Would you like to know that?
We just talked about a paper that took advantage of the haploid cell line. I'm curious as to what biological functions might have been missing.
I'm looking up the number of genes on the X chromosome,
Thank
okay?
You. Thank you. You can actually find that.
It looks like about 100,
100.
okay?
100.
There it is, right on the page here, you see? A
That's
through X.
Incredible. A through X.
So what has that got you that you now know that?
Well, because we have one of those in each one of our cells, thank goodness.
Hey, Rich Condit,
Yes, sir.
do you think any of the experiments you've done with vaccinia virus over the years, the outcome would be any different if you used a male versus a female cell?
I really doubt it. But
Yeah.
we're talking about a virus that's really
Just blows everything away,
yeah,
right?
Exactly.
No subtlety.
Yes, and not subtle at all.
That's right.
If you were a cell, about the worst thing that could happen to you is
It's a
vaccinia
pox-
virus. Yeah, it's a
that's
pox
right,
virus.
that's right.
So I think certain things wouldn't matter and others would. And preclinical in particular, if you're looking for the effect of drugs or vaccines or other
Sure,
therapeutics,
sure.
yeah, I think you should do both. And if you're doing an experiment in mice or dogs or hamsters, I think that you should balance the sexes in your experiment. I thought that was accepted practice.
It's not the case in parasitic diseases research. I can
Really?
tell you that. Yeah, because female mice, for instance, have a menstrual cycle. And they have hormonal levels that vary throughout the month. As the result, when you infect them with a parasite that's linked to those cycles, you get variations in the number of parasites or the take of the parasitic infection, etc. If you use male mice, you don't have those problems. So most of the studies that I was aware of using mice, at least, they were all male. And they were all 3-month-old male because that's a mature animal. So those were the gold standards that everybody applied. And you're right. I mean, we tried female mice, but the statistics were so daunting to try to make sense out of the results that everybody switched to males.
You know, well, I'm going to get in trouble here, but, you go into DNA replication, transcription, translation. I would think those are universal in in all cells, right?
Sure.
They're regulated in different ways. So now when you
Cells.
look at regulation, that could
Yeah,
differ, right?
But you're talking about cells in a culture dish. I'm talking about an entire animal.
Yeah,
I understand.
I think in the case of a cultured cell line, especially, you're dealing with something that is so far removed from
Exactly.
normal animal biology that I'm
it.
not sure saying it's a female or male cell line is going to be all that relevant.
Yeah, but still, the question's been raised, so I think it's an interesting
The question
one.
has been raised, and you could certainly imagine
Sure.
issues where it could come up. And by the way, I finally dug up a karyotype for HAP1 cells, and it looks like they have
good.
looks like
What do
they
you
have
got?
a Y chromosome listed in there.
Male.
Interesting.
Oh wait, no, they've got. Oh, I'm sorry. They've got an X.
They could
and
have.
the Y. The Y is missing.
Okay.
The Y is missing.
Okay.
Okay, so they appear to be female.
Sort of.
Sort of. Yes.
Half
Because they're
female.
haploid. Yeah, it's interesting. All right, we've done a half hour on 3 emails.
On 3
Yeah,
emails.
yeah, but still, no, these are good questions.
No, I love them. I'm not complaining. I just
These are
think
lateral wanderings.
I'll just read the last one. Then we should move on from John, who sends a link to Colorado Springs Rodeo canceled due to horse herpes. This is in the Gazette.
Straight from the horse's mouth.
or whatever.
So a rodeo has been canceled because one of the horses has tested positive for equine herpes virus. And this can cause problems apparently in horses. I was thinking as I was reading this that we should probably get somebody on to talk about. It's called equine herpes myeloencephalitis, EHV1. And for example, I don't know much about the biology of this. Is it become latently infected?
Yeah, I quizzed Dave Bloom about this.
What did he say about this?
He said it's really hard to find a seronegative horse.
Yeah, I'd figure they're all
This is
positive.
kind of like HSV1,
Yeah.
right? Where horses get it, goes latent. It's an alpha herpes virus, I believe. So it's yes, it is an alpha herpes virus. So it's latent in neurons, and it's latent in neurons sort of all over the body, and occasionally it can cause neurological disease of some sort. So there was on this article one comment that said that this canceling a rodeo because of this outbreak was a total overreaction because all horses have this anyway, and my guess is that that's probably correct.
That's what I was figuring. That it's like herpes in people, right?
Yeah,
You might say
exactly.
it triggered a knee-jerk reaction.
Well, I was
According
just
to
gonna
Roy
say, you know,
Rogers.
if your rodeo is canceled, you should get back on your horse and
That's
never mind.
The virus is spread mainly through nose-to-nose touching. That's what horses
They
like
do
to
that.
do.
They do that a lot.
It's transmitted on equipment, trailers, and water buckets.
Goodness.
Can show signs such as wobbly or uncoordinated legs and fever.
Whoops.
Well,
But most horses, I think, just get it and there's no problem,
Right.
and it goes latent. And
Except the horse that Lee Marvin rode.
yes, and,
can you be multiply infected? That's what I want to know.
Ah.
Can you be multiply infected? Because it sounds like these are older horses. They're in a rodeo, right?
right, they're getting somehow super infected or something like that. That I don't know.
Yeah, because it sounds like they're getting super infected, and then in that case, they may not be. It's a little different from human herpes, right?
Wait, rodeos use older horses?
Don't they? Well, they're not young. They're not brand new. They're not foals, right?
Some are
Right, but
3-year-olds
I
and
assume
4-year-olds.
They weren't like headed for the glue factory. I mean, they've got to be
wait,
pretty active
So
to be in
this,
a rodeo.
I don't know anything. What's an old horse?
I'm sure that there is a veterinarian out there.
Who's freaking out?
That's right.
Wait,
Well,
how long do horses live?
A long time.
They usually
Come on,
live
Dickson.
until they die.
No,
but
20,
sometimes they don't live
25
that
years.
Okay, so a rodeo horse is what, 5, 6, 7 years old?
It's possible.
All right, what I'm saying is by the time you're a year old, if you're living in a barn, you're probably going to be infected by other horses. So
Probably.
all these rodeo horses are likely to be infected already.
If you're a horse.
The question is whether you can be reinfected and show disease symptoms. That's my question. It says in this article that there is a vaccine, but it's of questionable efficacy.
Yes, and Dave said the same thing. That the vaccine is of questionable efficacy.
Here.
I mean, my assumption is that the symptomatic horses are not getting that. It's a reemergence from latency,
Right.
as with HSV1. But I don't know that for a fact.
Well, and one of the, I guess, riders' ears saying every horse contains the herpes virus.
Yeah,
every
yeah.
horse. You get it active by stressing the horse out,
Which a rodeo does, right?
which I suppose
Right.
a rodeo would do.
Right. And I'm really glad that one of the rodeo riders understands that.
Yeah,
Yes.
that's good. That's really good.
That's good.
By the way, speaking of horses,
Yes.
as you know, the Hendra virus emerged in horses in Australia
Right.
in the 90s. And,
Right.
well, I was there. I met the man who isolated the virus.
Oh, cool.
He was in the BSL-4. He said on a Friday he took some horse clinical specimens and inoculated some Vero cells. And then Monday morning he came back and they were all dead.
Wow.
Wow.
He had it there. So they actually have a vaccine that's used in race horses.
Yes.
And it prevents them from getting infected from the bats and protects people because that's how people get the infection. So you don't need to immunize people in that case.
Right.
The horses are race horses, which are very expensive.
And that's a big deal in Australia. It is.
What is a big deal?
Racing horses.
I wouldn't know, Dickson.
Well, it's a big deal everywhere it's done, I
It's
think.
true.
It's pretty big here, right, Dickson?
It's a big
Very
deal in
big.
New Jersey, I know that.
Very big.
And Baltimore.
And Baltimore. That's true.
Okay, let's do some picks, Dickson.
I'd love to.
Do you have one?
I do.
What is it?
I sent it to you.
That's not an answer.
It doesn't have a name. I couldn't find a name for this. It's a
just
YouTube.
called
It's a
sci-
YouTube.
It's called Science Fun.
Oh, but of course it's science fun, but there's a series of them. This is the one I picked though. It's a vibrating frequency on a metal plate on top of which is a non-sticky powder, which follows the contours of the vibrations throughout this material that it's sitting on. And as the frequency increases or gets more compressed.
Well, it increases the frequency is correct.
Yes, I meant to say that. I did.
The
say
wavelength
that,
gets
as
shorter.
the wavelengths get shorter, and the noise gets higher in pitch.
Make different patterns.
The pattern on the surface
cool.
of the plate changes, and watch how it changes. It ends up as an extremely complicated deal that you would never expect by looking at this piece of flat metal that just looks like nothing.
This piece of metal is called a Chladni plate, by the way.
Chladni plate.
CHLADNI.
And what? And Alan, you're going to tell us exactly what a Chladni plate is, aren't you?
Yes.
Oh,
So
good.
Chladni was a physicist. I think he was 19th century or something.
Okay.
And he came up with this method. We used to have some of these at the Franklin Institute in
Oh, is that
Philadelphia
right?
when
Yeah,
I
yeah.
volunteered there.
Oh, neat.
And the way we would do them at the Franklin was we had a violin bow.
Right.
And you've got this plate that's sitting there.
What's it made out of? What is it made
It's
out of?
metal. It's just a piece
okay,
of, I think they
okay.
were steel.
Why is it called a Chladni plate? Why not just a
Because
piece of
Chladni
steel?
was the guy who invented it.
Ernest Chladni.
But it's
Ernst Chladni, that could very well be.
Is it a
Yes.
special piece of steel or just a piece of steel?
It's just a flat piece of steel with a post attached in
Okay,
the center
okay.
of
Yeah,
it.
look at this. It's on Wikipedia.
Oh, that's nice.
CHLADNI.
There you go.
Nice.
And you know what? I'm going to put this in the comments to
Oh,
this video because all
please
the other
do that.
comments are just moronic.
Yeah, so what we did, this I think is exciting it with a like a speaker.
Exactly.
The traditional method, which is what we did at the Franklin Institute, was you take a violin bow and you stroke it against the side of the plate like playing a saw,
Got it.
and it would make a sound, and you throw a little sand on top of it, and you'd get a pattern.
It's amazing.
Yeah.
I was stunned by the complexity of the pattern.
Would high school or elementary school kids learn anything from this, Dickson?
Well, yes. The one thing they would learn, if nothing else, is even if they can't explain the pattern, they would have never predicted that by looking at this simple object.
Okay.
So from simple things, complexity arises.
Well, and
that's-
you can make the point that sound is in a wave.
Indeed, you can. And
Yeah.
what did you say in the Franklin Institute when you were
Well, it was
demonstrating
part of a
this?
whole display on waves.
Okay.
And so we would
Okay.
demonstrate. I would demonstrate the Chladni plate and say, that the waves of sound are making these patterns, which
Right.
form these wave-like patterns.
Right.
And then, if you look over here, this machine is demonstrating how a
Okay.
wave
Okay.
propagates.
Okay.
And yeah,
Alan, what do you have?
I have a Flickr page from somebody named Nikki Bay, who does macro photography of insects. And I know I've picked a bunch of stuff like this in the past, and I'm going to continue picking stuff like this when I come
this
across
is
it.
awesome stuff.
Yeah, because this guy's stuff is really cool. And he's just, I think he's an amateur at this. This is just his hobby. He goes out and
it's cool.
He's in Singapore or Thailand or something. Singapore.
How beautiful.
So he goes out and shoots bugs on the weekend.
That's amazing.
And does a beautiful, beautiful job of it.
And he's extraordinarily prolific.
Yes.
I love
there
it.
are hundreds, if not thousands, of these pictures. It's
11,660 photos, it says on there.
Dixon, you better get going.
I better get going, yeah. Alan, I'll send you a few of my own.
Okay.
You won't want to post
Why don't you
them.
have a Flickr page, dude? No, it's you're too old school, right?
That's right.
That's gorgeous. Look, I like him with that setup he's got, taking a picture on the sand there.
It's lovely.
Yeah.
What are those, 2 lights or something?
Yeah, he's got a flash that he's rigged up on the lens because lighting is...
Yeah, they use a circle
That's pretty cool.
flash.
I like
usually.
that.
It's wonderful. Great pick.
Rich Condit, what do you have?
Let's see. What do I have? Yes, I picked I just ran across this. FASEB has had bioart competitions in the past, and they've just announced their 3rd annual bioart competition. So this is a competition where if you do something in your science stuff that you do that you think looks cool,
Sure.
doesn't matter what it is. It can be, pictures of or patterns of any sort. You can submit it for the bioart competition. The deadline, I believe, is August 30th. So there's still time, gang.
There is.
Actually, one of the winners from last year was somebody here at UF. I just realized
Get out of here.
looking at this site, and so it's cool.
Dixon, can you enter this?
Good.
Why don't you do it?
Wow.
Yeah.
You have to have something to enter first, don't
You
you?
have pictures of bio.
I do.
Pictures of bio.
Pictures of bio.
Need to get you going here, Dixon.
You've gotten me going already.
All right. My pick is a podcast called Up Close. When I was in Melbourne, I discovered this podcast. I was actually a guest on an episode. It's quite a different podcast from ours. First of all, it is supported by the university.
Right.
They have a studio that they use, which is very nice.
Right, nudge, nudge.
And they have not just a couple of hosts, but they have a technical staff there and lots of people that help out.
Right.
And it's a bit different from ours because they cover all sorts of fields, not just science, but politics and what else is there? Law and finance and everything else. So, they have a couple of hosts that can cover everything, but it's not a conversation like ours. There's a host and myself, and she asked me questions, and I answered, and so there is a place for that, and it's quite well done. They do a good job, and so she talked to me about polio and that sort of thing. But there's some other episodes there that you might like as well. So check it out. Up Close, research, opinion, and analysis.
Cool. Lovely.
Give them a TWiV bump. And we have a couple of listener picks. One is from Mark Martin, who's a microbiologist over at the University of Puget Sound. Does that sound right?
Yeah.
Is there a University of Puget Sound?
There is.
It's an undergraduate college, and he teaches
Tacoma?
undergrads about microbiology. I think so. And I know him. I've met him at meetings. And on Twitter today, he wrote, "For all you cartoon bacteriophage fans," PHANS, I found this. And it is a website called The Average Bacteriophage, the infamous virus and its normal life. So he was just pencil drawings of phages doing different things.
This is almost like, this reminds me of elephant jokes, basically.
Yes.
I mean, this is...
Bacteriophage getting its daily workout.
Bacteriophage unable to sleep because of the turbid dance party next door.
Ooh.
It's pretty funny. There's simple drawings, but I like when you focus on phage. Bacteriophage confessing its sins to a priest phage with a cross on its tail thing.
Right.
It's cool. So thank you for that, Mark.
Our knowledgeable virus who remains unphaged by anything.
Oh, that's good, Dickson. Very good. And another from Conrad. Hiya, TWiV. Just a listener pick for you. Scholarpedia, the peer-reviewed encyclopedia. I was wondering if you've heard of it and if you're interested. Interested in what?
Exactly.
Right now, there aren't nearly as many topics as on Wikipedia, but it looks promising to me and might be worth keeping an eye on. Bye for now.
Is that an ion? Is that a divalent ion or a...
Knowledge is curated by communities of experts.
It's like Wikipedia, except the articles are actually peer-reviewed.
Wow.
It is more closely scrutinized by individuals who are credentialed in the relevant areas. Okay.
And that's why there are fewer than Wikipedia, of course. The areas are quite narrow at the moment.
At the moment.
Yeah, so anyway, give it a TWiV bump. There's no nothing like life sciences as far as I can tell here.
No, I looked around for viruses and et cetera. They've astrophysics, celestial mechanics, computational science, computational intelligence, dynamical system physics. There's a section on touch.
Lord.
It's based on who they had handy when they started the site.
Yeah, exactly.
Touch,
right,
handy. Thank
Theoretical
you.
high-energy physics, space-time and gravitation. Here's an encyclopedia of play science.
Really?
Oh, that sounds fun.
Yeah, that should
There you
be
go.
good. There you go.
Serious stuff, guys. Come on.
All right, thank you, Conrad. That'll do it for TWiV 295. You'll find it at twiv.tv, iTunes, and do send us your questions and comments to TWiV at twiv.tv. We will get to them. We promise. It just can take some time, but we
As
will,
you've seen, it
we
can take
will read them all.
That's right.
Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
Welcome.
Seem to be engaged today.
Had a lot of
I'm
fun.
just kidding. That's a jib. That's a jib jab, you know.
You just woke me up. I'm sorry. What did you say?
Dickson, are you going fishing this weekend?
No, I'm not. No, I'm not going to go fishing until I get my new glasses.
Well, you think that's going to make a difference?
It's going to make a big difference. I can see the flies.
Okay.
You have to be able to see those flies to fish them.
You're going to Patagonia, aren't you?
No,
Where are you going?
Peru.
It's the same thing, isn't it?
No.
I just insulted all the
No.
Peruvians and the Patagonians.
Patagonia is much further south.
Okay. Have fun. Well, you're
I'm
going to
gonna-
be here next Friday?
I'll be here next Friday.
Okay.
Sure.
Alan Dove is at turbidplaque.com. You can also find him on Twitter as Alan Dove. Thank you, Alan.
Always a pleasure.
Thank you, Alan Dove. I didn't know you taught high school while you were a PhD student here.
Yeah, I volunteered at, teaching speech and debate like twice a month or so.
Yeah.
I'd go to a high school, Urban Peace Academy, inner city high school. That
Nice. Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello, and I'm at virology.ws. And this is, episode 295. In 5 more episodes, it's number 300. We're all going to be in Washington, D.C., and we're going to do a live episode. We're going to have an audience, and we'll give you more details next time. But
Excellent.
Check it out. We'll all be together in a room doing TWiV for the first time in a room together, right?
That's right.
I don't know how to quite say that, but...
that came out okay. People would get it.
That's
It may
fine.
have to untangle it a little bit.
Have
we'll all
we-
be in the same place.
Have we been in the same room, all 5 of us?
I've never met
No.
Kathy
Never.
Spindler.
Okay, so that's one way to put
I'm
it.
looking forward to it.
But Alan met Kathy in Boston for
I
the
met
first
Kathy
time.
in Boston. Yes,
Yeah.
I've met everybody on TWiV in person, but not all at once.
All at once. We'll be all at once, and there'll be
Yeah.
an audience, and we'll be live streamed.
Should be fun.
So we will prove to the world that this is not just one really gifted ventriloquist.
That's right. Final proof as if anyone needs it, I guess.
Exactly.
Right? You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.